Thank you for the invitation to present today, and I've been asked to present on test validation in medical laboratories from an Australian perspective. And so I'll give some context as to where I'm based and what we do first. So I am based at the Garvan Institute of Medical Research, which is in Sydney. It's a beautiful part of the city, you can see from this image, and I've highlighted some of the affiliated institutes and hospitals with the Garvan Institute of Medical Research. We have St Vincent's Hospital, the Kinghorn Cancer Centre where I'm based, and the Garvan Institute, as well as a medical school nearby and the Victor Chang Cardiac Research Centre. So at the Garvan Institute there are around 600 scientists and clinicians, but there are more than 1,000 scientists and clinicians across the precinct. So it's a very rich grounds for collaboration and for clinical projects. The lab where I'm based and that I'm director of is at the Kinghorn Centre for Clinical Genomics, and our lab is the largest DNA sequencing facility in Australia. My experience in research and clinical testing spans around 20 years. As a cancer researcher, I studied the genetics and epigenetics of hereditary and sporadic bowel cancer. I was a group leader at UNSW starting 2009. Before that, I was in the UK. And then since leaving UNSW, I've been in leadership roles in genetic pathology, first in a private pathology company known as Douglas Hanley Moyer Pathology. Those of you in the US may know this as Sonic Healthcare. The headquarters of Sonic Healthcare is actually DHM in Sydney, and I was the supervisor of the genetics lab there. And since then, I've been laboratory director at the Kinghorn Centre for Clinical Genomics Sequencing Lab. And in that time, I've overseen the validation and NATA accreditation of multiple single gene, gene panel, whole exome, and whole genome services. So that just brings my expertise to highlight why I've been asked to give this talk and hopefully shed some light on what test validation for clinical testing is like in Australia. My lab is an ISO 15189 accredited lab and also NATA accredited for germline whole genome sequencing in 2016, germline whole exome sequencing in 2018, and most recently somatic gene panel testing in 2020. So ISO 15189 is a set of Australian or international standards actually for operating clinical laboratories, and it builds on other standards such as ISO 17025, which is focused on technical requirements, but the ISO 15189 is really focused on clinical safety, so how samples are handled within a clinical lab. Now, when it comes to NATA, this is an Australian-based organisation, and so let's go into these in more detail because it's really important to understand what these are because they guide how we validate tests in a clinical lab. So NATA is the National Association of Testing Authorities. It provides administrative support. It provides the assessors to assess a lab to determine whether they should and could offer clinical testing, and it provides the infrastructure around those. The Royal College of Pathologists of Australasia, they provide professional advice, training and support and work hand in glove with NATA. And NPAAC, the National Pathology Accreditation Advisory Council, are a ministerially appointed council which basically provide the requirements that a medical laboratory must follow in order to perform clinical testing. And NATA will assess a lab based on the requirements that NPAAC publish. NPAAC have a series of standards, documents separated into tiers, which are used as a guide to validating new tests and operating clinical lab, and I've listed them here. So ISO 15189, as I mentioned, is an international standard that outlines the requirements for quality and competence for medical laboratories who perform testing in clinical or diagnostic settings. It's focused on the clinical and technical validity, patient safety, and risk reduction of any operation within a lab. And as I mentioned, there are other standards that this builds on, such as ISO 17025, which is more focused on technical competence. But in addition, the ISO 15189 is focused on management system requirements that are necessary for it to deliver testing in a clinically and technically valid way. And what does ISO 15189 consider? Everything from pre-analytical steps such as sampling, handling and transportation of a sample, the validity and appropriateness of a method in the lab, traceability of measurements and calibration to national standards, suitability of those pieces of equipment for testing, the technical competence of staff, which should be documented and recorded and continually improved, the quality management system, and also steps that the lab must take to continually improve what it offers. And some of that involves customer feedback and making sure that complaints are addressed. So when it comes to validating a new test in an Australian medical laboratory, any test that is developed from scratch or an off-the-shelf test that is modified in any significant way is known as an in-house in vitro diagnostic medical device, or an in-house IVD. The US equivalent would be known as a laboratory developed test, or an LDT. And again, there are requirements that must be followed in order to validate a test for clinical use. As I mentioned, NPAAC, but also the Therapeutic Goods Administration, and these are two documents that are Bibles, I guess, for medical laboratories who wanted to validate a new medical test. In terms of managing a lab, there are certain pragmatic factors that guide what test would be validated, and everything from the workflow and the equipment used. Some of those pragmatic factors include the scalability and automation of a method. You want to reduce hands-on scientist time where possible. It may be a test that's amenable, or it may not be amenable to automation, and some of those steps may involve liquid handling, robotics, LIMS integration, the automation of analysis with bioinformatics, but also you have to factor in what the cost of that automation is. You also have to factor in clinical safety and whether the test can be operated with clinical safety in an ISO 15189 environment. Turnaround time of the test is important. It has to be guided by the urgency of clinical management. It makes no sense to have a test that takes 4 weeks to perform, but in order to guide clinical management, a clinician may require a test result in 3 days. So that's an important factor as well. And of course, the cost. Cost as in what's the COGS, the cost of goods for the lab, whether the market would be willing to pay the cost, and also in the Australian context, whether there's Medicare rebate available that would cover costs. When it comes to test validation, there are a number of specific criteria that we need to address. And this is standard across the world, nothing too specific here in terms of the Australian context, but we would want to make sure that the test is accurate, is robust, precise. We need to define the measurement uncertainty of any quantitative measurements, and we need to understand the limitations of the test and whether there are any potential causes of false positives or false negatives. And I've defined what those terms mean here. So rather than go through a series of standards and documents that are quite dry, necessary but quite dry, I thought instead what I would do is go through my personal view of test validation in a medical laboratory. And I'm approaching this in this slide from the perspective of a researcher. And having worked in a research environment, I'm aware of the way or the disconnect between researchers and research projects and a lab that performs medical testing in a regulated environment. Now, a researcher often thinks along these lines. We have an idea, does the test have clinical utility? And along the way, you would design a method that would be able to measure, for example, mutations in a particular gene. You would then validate it, and then you would have a test with clinical utility. I think this is the wrong way of going about test validation. It really doesn't account for the many factors we need to take into account when designing a new diagnostic service. And that's really where we want to get to, a diagnostic service that has clinical utility, that is clinically accredited and can be used by clinicians to inform patient management, that is profitable or at least has its costs covered, is scalable so that if you receive 50 samples in a year, you're fine, but if you receive 2,000 samples in a year, you can cope. That is clinically safe, quick, with an appropriate turnaround time for the clinical question being asked, and is also reliable. So to get to that point, yes, the test has to have clinical utility. There also needs to be a business case, and as I go through these slides, I'll define in greater detail what each of the factors and the considerations for each of these. Then you design your method. You also need reference samples to test during test validation, which you then perform, and I'll come to the steps there. Your laboratory needs to enroll in quality assurance programs, and this is proficiency tests, external proficiency tests, to make sure that your test is detecting what it should. You then need to resource the test. That may involve training the scientist to perform it and the pathologist to interpret it. And then you need to market the test to make sure that people know that you have developed this and target that towards the most appropriate clinicians. So let me give you a case example. In this case, I'm going to describe the genetic testing of a gene called UGT1A1. UGT1A1 encodes an enzyme that conjugates bilirubin in the liver. And if it's not functional, then you end up with the accumulation of bilirubin in the blood, or hyperbilirubinemia. This is also known as Gilbert syndrome, but certain genotypes can also be predictive for adverse reactions to a range of drugs, indinavir, atazanavir, and irinotecan. Indinavir and atazanavir are used in the treatment of HIV, and irinotecan is used in the treatment of gastrointestinal cancers. If given these drugs, and you have Gilbert syndrome, you'll have toxic side effects to those drugs. So it has clinical utility. But is there a market for it? This is where the business case comes in. You need to understand whether people will request this test. You need to understand whether anyone else already offers it, and whether your test is predicted to be more economical, have a superior diagnostic yield, be more efficient, be safer, and be quicker, or have a decreased turnaround time. The method design in this case can be a simple PCR and capillary electrophoresis-based method. So that's great because it's quick, it's simple, it's easy, it's cheap. Now, the genotypes that are diagnostic for Gilbert syndrome and predictive of adverse reaction to specific drugs are all located within a specific region of the UGT1A1 promoter. It's a dinucleotide repeat, and there are only 4 clinically actionable variants that we need to consider. And if you're familiar with the pharmacogenetic system of haplotype calling, then you'll be familiar with the star allele system, and they are known as star 1, star 28, star 36, and star 37. So as I mentioned, these variants are all dinucleotide repeat lengths, and they're all within the same location. So a simple PCR capillary electrophoresis assay and only 4 genotypes, the number of samples we would require is quite small, only 20 to 30, with representative examples of each of those genotypes. Obviously, if you were doing gene sequencing across the entire gene or a panel of genes or a whole genome, you would require many more reference genotypes to be assured that you're detecting the full spread of different types of alterations that you may see. Then comes test validation. It will include a period of test optimization, but then you get on to testing the specific criteria of accuracy, robustness, measurement uncertainty or precision, the limitations, and then documenting a validation report which will be submitted to NATA in the Australian context. The quality assurance program step is really important because it's a regulatory requirement that a lab involved in medical testing is enrolled in some form of proficiency testing. And as I mentioned before, this is really around making sure your test detects what it should detect and defining the false positive and false negative rate. And that's also crucial for understanding the limitations of the test. Now, if there's no quality assurance program available, then the next best option is to set up a sample exchange with a laboratory that performs similar testing. And we've had to do that in the past as well. Then you need to resource the test. You need to make sure it's scalable, cost the pathologist time, the scientist time, standardize where possible and automate where possible, and that may involve the reporting as well as the wet lab work. You need to make sure that you have the right number of scientists with redundancy in personnel and equipment, that is. And you need to consider that the other services within your laboratory aren't affected by this new service. And that could be in the resourcing context, but also in the methodological context. Is it going to require equipment that would displace other important equipment? Are there contamination risks? For example, an amplicon-based test next to a non-amplicon-based test, and other considerations such as those. Then you need to train your scientists. In the regulatory environment, you need to make sure that those training exercises are documented and that there are records for each member of staff who will be performing the test. And then finally, marketing to your target audience with mail-outs, flyers, presentations. I often have to go and give presentations to show clinicians and oncologists a new test that we've developed, to explain to them the clinical utility, the test request route, and how this compares with other tests, similar tests that are available. So once you've done all that, you submit a validation report to NATA. NATA will then, if it's a completely new scope of service, a scope of accreditation that you're applying for, then they will visit personally with technical assessors and a NATA lead assessor, and they will go through with a fine-tooth comb all of the processes that you've set up that test uses. Everything from your quality management system, training records, SOPs, the validation report itself, the way you store samples, the way you return results to requesting clinicians, and then they deliberate and get back to you within a few weeks to potentially a few months for a complicated extension of scope. Once you've been awarded accreditation, there's then reassessments and surveillance visits. And the surveillance visits are usually conducted every 12 months following initial assessment. But also, if you have recently changed the way your lab operates or your service, you may get a surveillance visit earlier than expected. And then reassessments are conducted every 24 months. So there are other important documents as well. As I mentioned, there's the ISO 15189, ISO 17025, the NPAAC tiered documents, some of which are listed here. And there's also published literature describing standardized frameworks for the validation and verification of clinical tests. And there's an example here by NATA et al. And with that, that's the end of my presentation, and I'll happily take any questions. Thank you for your attention.